Good day. So this is my lecture for chapter 9, which is senses. So to start, what is senses? So a sense is the ability to perceive stimuli. So we also have what you call your sensation, which is the consciousness awareness of stimuli received by sensory neurons. So we also have what you call your sensory receptors. These are sensory nerve endings that respond to stimuli by developing action potentials. So in this diagram, you will see how the senses are being um, categorized. So in senses, you have receptors distributed over the large part of your body, which what you call are your general senses, and you also have senses wherein the receptors are localized within the specific organs or special senses. For general senses, you can have the locations in your skin or muscle joints, which can be somatic, like touch, pressure, proprioception, temperature, pain. And some can also be located in your internal organs or the visceral part of your body for pain and pressure. And then for special senses, we have the sense of smell, taste, sight, hearing, and of course, balance. So the types of senses includes general senses. These are receptors over large part of the body that sense touch, pressure, pain, temperature, and itch. So the somatic provide information about body and environment, and the visceral provide information about internal organs. And then again, special senses are for smell, taste, sight, hearing, and for balance. So for the types of the receptors, first we have what you call your mechanoreceptors, which detect movement like touch, pressure, and vibration. You also have what you call your chemoreceptors, which detect chemicals like odors. You also have photoreceptors that detect light. You also have the thermoreceptors that detect temperature changes. And you have the nociceptors that detect pain. So for the types of your touch receptors, we have first your Merkel's disc, which detect light, touch, and pressure. You also have your hair follicle receptors that detect light, touch. And you have your Meissner corpuscle that is found deep in your epidermis, and it is for localizing tactile sensations. Next, you also have your Ruffini corpuscle, which is a deep tactile receptor, and it detects continuous pressure in your skin. You also have your Pacinian corpuscle, which is the deepest receptor associated with tendons and joints. It is also for detecting deep pressure, vibration, and position. And again, we have already discussed this when it comes to the parts of your skin, right? So these different types of receptors are actually embedded in your skin layers. So first, let's discuss, we have here your free nerve endings. So these are responsible for painful stimuli, temperature, or itch. Okay. So here is your free nerve endings. And then you have your Merkel discs. So they detect light touch and superficial pressure. Next, you also have your hair follicle receptors, which detect light touch. And then you have your Meissner corpuscles, which are involved in fine discriminative touch. And then you have your Ruffini's end organ or the Ruffini's corpuscle, which detects continuous touch or pressure. And then the Pacinian corpuscle for deep detection of pressure, vibration, and for position. So for pain, this is actually an unpleasant perceptual and emotional experience, and there are two types. It can be localized, like sharp, pricking, cutting pain. It has rapid action potential, or it can also be diffused, like burning, aching pain, and slower action potentials. So for pain control, you have your local anesthesia. Its action potential suppressed from the pain receptors in local areas, and chemicals are injected your near sensory nerves. So usually these local anesthesia are like being applied when you visit the dentist and you need to uproot a tooth, okay? So pag local anesthesia, kung saan lang yung part na kailangang gawing numb or maging paralyzed, dun lang nilalagay yung local anesthesia. But when you say general anesthesia, it would actually cause you loss of consciousness, and the chemicals affect your reticular formation. So ito talaga yung mga ginagamit when it comes to surgeries, no, na talagang pinapatulog ka so you won't feel the pain. for during surgeries, and of course for yung mga nagpapa-opera kapag kunyari sa lower parts of the body, kung may specific parts of the body na kailangang operahan or galawin. Okay, so yun yung mga ayun yung ginagawa ng general anesthesia. So next you have your what you call your referred pain. So what is it? It originates in a region that is not source of the pain stimulus. So it is felt when internal organs are damaged or inflamed. So you also have what you call your sensory neurons from the superficial area and neurons of the source pain converge onto same ascending neurons of the spinal cord. So yung referred pain, 'di ba? Parang yan yung kumbaga minsan ginagamit to ng mga 'di ba, sa mga Chinese kapag kunyari meron kang gustong ipagamot na part of your body or ipahilot na part of your body, 'di ba, sa foot, 'di ba, may tinatawag silang mga foot reflexes. So kumbaga ganun yung idea of referred pain. So hindi porket kumbaga ibang part ng katawan mo yung sumasakit, doon na nagmumula yung pain na yon. Minsan it comes or it is originating from a different part of your body. So for example, the areas of referred pain in the body surface. So the pain from the indicated internal organs is referred to the surface as shown. So for example, if you have pain in your liver and your gallbladder, usually it affects the part of your shoulders. Okay? Pag lungs and diaphragm naman, dito sa may bandang leeg. So for the stomach, it is given that it should be somewhere sa gitna ng tiyan. Okay? So hindi sa lahat ng pagkakataon kung saan yung masakit, doon mo na malo-localize yung or doon mo na sasabihin na doon nang gagaling yung pain. Okay? So it's not all the time. Pero most majority nung mga merong referred pain, talagang doon kumbaga doon nadadiagnose na may problema talaga. Like example would be your colon, your kidneys, 'di ba, your appendix. Usually sa mga parts talaga na 'yan, nagkakaroon talaga ng idea na may problema. Okay, so the confirmatory na lang dyan is for you to get further test para ma-confirm na ito may problema talaga siya. Okay? So next, let's go to olfaction. So what it is the sense of smell. It occurs in response to the odorants. The receptors are located in the nasal cavity and hard palate, and we can detect around 10,000 different smells. Okay, so in this one, in the image that you see here, you see that you have your your palate here, you have your nasopharynx, you have the nasal cavity, and then on your olfactory recess, on it, you will see your olfactory nerves. Okay, and then you have there your olfactory bulb, and then the olfactory tract that would eventually go to your brain. Okay, so that's how your olfactory nerves look like sa inyong ilong. So how does olfactory work? So the nasal cavity contains a thin film of mucus where odors become dissolved. Okay, and then olfactory neurons are located in the mucus. Dendrites of the olfactory neurons are enlarged and they contain cilia. Dendrites pick up odor, depolarize, and carry odor to the axons in the olfactory bulb or the cranial nerve 1, and the frontal and the temporal lobes process what type of odor you're smelling. Okay? So this is actually how smell is being perceived. So first, you have here on the mucus layer of your epithelial surface, you will see now the ends of your cilia. Okay, so the ends of the cilia, you will see there now the dendrites. So this is already the olfactory epithelium. In the olfactory epithelium, you will see your dendrites coming up, and then your olfactory neuron. So going to then passing by the connective tissue, you have your axons, which help form the olfactory nerve. And then from the passing again your cribriform plate, you now see the olfactory bulb and the olfactory tract. So you see there the interneurons in between, so the axons do actually connect with the olfactory tract. and of course the other interneurons that are associated when it comes to processing the smell. Okay, so from cilia going up to your olfactory bulb, and then the messages from the axons going to the interneurons will go eventually to the olfactory tract going to your brain. And then your frontal and temporal lobes will decode or will analyze what type of odor you're smelling. Okay. So next, sense of taste. So for taste, we have taste buds, which are the sensory structures that detect taste. They are located on the papillae on the tongue, the hard palate, and the throat. So inside each taste bud are 40 taste cells. So that is definite. So inside a taste bud, there should be 40 taste cells. So each taste cell has taste hairs that extend into the taste pores. Okay. So here you see the image of how the tongue looks like. So you have here the surface of the tongue, and then you have the papillae. At the back part, you see the palatine tonsil, the linguinal tonsil, the root of the canal, and then your epiglottis. But if you see on, if you check on the papillae or the taste buds, no, this is actually how it looks like. So you have here the epithelium, the top, the taste bud, and then you have your papilla. So much smaller image shows that this is actually how it looks like. So you have here the taste hair sa bukana. Okay, you have the taste hair here. And then you have your taste pore, where your taste hair protrudes. And then, pagpasok sa taste pore, from the taste hair, you will see the taste cell. Okay, each has the nucleus. Okay, taste cell. And then from the taste cell, magko-connect na siya with the nerve fiber of the sensory neuron. Okay. So, apart from having the taste cell, you will also see supporting cells around it. And of course, you see there your epithelium where your taste hair and supporting cells are embedded, going to the nerve fiber of the sensory neuron. So, how does the taste work? So, taste buds pick up taste and send it to the taste cells. So, taste cells send taste to the taste hairs. And then taste hairs contain receptors that initiate an action potential which is carried to the parietal lobe. And then the brain processes taste. Okay? So the types of tastes, you have sweet, sour, salty, bitter, and the last is umami. Certain taste buds are more sensitive to certain taste, and taste is also linked to smell. Di ba nga kapag may someone na nagluluto ng ulam at pag naaamoy nyo, minsan parang nalalasahan nyo. Diba? Minsan din kahit mabaho yung amoy, nalalasahan nyo kung paano yung lasa ng amoy na yon. Okay? So, basically, the nose and the tongue or the mouth is connected when it comes to taste. Okay? So here, you will see the different parts or the different types of papillae found in the surface of your tongue. So first, you can see here the fungiform papillae. Okay, and then the filiform papilla. You also have the foliate papillae at the sides, and then you have the circumvallate papillae that borders the anterior and the posterior parts of the tongue. And then you will see the palatine tonsil at the back and your linguinal tonsils. So, the image on the right would actually show you which part of the tongue do decode the specific taste. Okay, so for the umami, majority of the tongue. For the sour, most likely the sides of your tongue. For the sweet, the tip. For the bitter, the back part. And then for the salty is the tip border of your tongue. Okay. So this is actually how the taste or the action potentials of perceiving taste look like. So if you have the tongue here, it is innervated by your cranial nerve 5. You also have 7, 9, and 10. So on the anterior part of your tongue, you will see that it is innervated by 5 and 7, cranial nerve 5 and 7. The middle part of your tongue is cranial nerve 9, and then the back of your tongue would be cranial nerve 10. Okay, so those are what you call your facial nerve. You have your facial nerve number 7, trigeminal nerve 5, glossopharyngeal nerve 9, and then vagus nerve 10. Okay, so from there, so once this is already perceived and once this cranial nerves already receive the message, okay, it will be directed now to the foramen magnum, okay, of your spinal cord. Okay. And then signals will be sent to interneurons, going passing to your brainstem and then going to your thalamus, and then going to the taste cortex or taste area of your cortex. Okay. So next for the vision, so you have accessory structures for the eyes. So first you have your eyebrow, which protects from your eyes from sweat and it shades from the sun. You also have your eyelid or eyelashes, which protects from foreign objects and lubricates by blinking. You also have what you call your conjunctiva. This is a thin membrane that covers the inner surface of the eyelid. Okay, so for the conjunctiva, you can see that you have the palpebral conjunctiva at the top, and then you also have a bulbar conjunctiva at the bottom. Okay, and then next you have the lacrimal apparatus. So the lacrimal apparatus, it actually is responsible for producing the tears, and your lacrimal apparatus includes the following parts. You have the nasolacrimal duct, which runs from the your nostrils, and then you have the lacrimal sac near your lacrimal canaliculi, and then at the top of kumbaga, lateral upper right or upper left side of your eye, you will see the lacrimal gland, and of course the lacrimal ducts. So this lacrimal, paano ba napupunta yung tears natin, no, dun sa canaliculi, malapit sa canaliculi, or the tear where your tears fall, di ba? Imagine this lacrimal gland is found at the upper right or upper left side of your eyes. So this lacrimal ducts actually travel across your eyes sa back, sa likod. Okay, and then may butas, di ba, dito sa may bandang mata. Okay, so diyan lumalabas yung tears. Okay? So, kaya din kapag ka umiiyak, automatic sinisipon. Kasi yung tears na nanggagaling sa lacrimal duct, pupunta siya dun sa side, papunta sa nasolacrimal duct. So, yun. Kaya nagtutubig din ang ilong kapag ka umiiyak. Kasi magkakakonekta sa lacrimal apparatus yung nasolacrimal duct and the lacrimal glands. Okay? Next, you have the intrinsic eye muscles. Of course, they help the movement of your eyeballs. So for the extrinsic and intrinsic eye muscles, here you have the different muscles of the eye. First, you have the lateral rectus. So this moves the eye laterally. It is controlled by your cranial nerve number 6 or the abducens nerve. You also have the medial rectus, which moves the eye medially. And then you have the superior rectus, which elevates the eye and turns it medially. And then inferior rectus, that depresses the eye and turns it medially. Inferior oblique elevates the eye and turns it laterally. And then superior oblique depresses the eye and turns it laterally. So it is being controlled by, more of it is the oculomotor or cranial nerve 3, but with also with the participation of abducens and the trochlear nerves. Okay, so please be familiar with the different muscles. Again, you have the lateral rectus, this one. And then you have the medial rectus. Okay, so your medial rectus is here. And then you have the superior rectus, this one, the superior rectus. And then the inferior rectus. Oh, this one is the inferior rectus, this one. And then you have the inferior oblique, okay, this one. And then the superior oblique. Okay, insertion of the superior oblique muscles. Okay, so those are the extrinsic and intrinsic eye muscles. So, for the anatomy of the eye, of course, the eye is a hollow fluid-filled sphere. It is composed of 3 layers or tunics, and it is divided into chambers. So the first one, or the outermost layer or tunic, would be the fibrous tunic, which has what you call your sclera. Okay, so it is a firm white outer part. It helps maintain eye shape, provides attachment sites, it protects internal structures of your eyes. And then you also have the cornea, which is the transparent structure that covers the iris and pupil, and it allows light to enter and focuses the light. Next, you have your vascular tunic, or the middle layer. It contains your blood supply. So it has what you call your choroid, this one. So it is black part, or because of the melanin, it delivers oxygen and nutrients to the retina. You also have your ciliary body. It helps hold your lens in place. And then you have your suspensory ligaments that hold the lens in place. Okay. And then next you have the lenses. Okay, so the lens is the flexible disc. It focuses light onto the retina. You also have the iris. The iris is the colored part. It surrounds and regulates the pupil. And then the pupil, it regulates the amount of light entering. So lots of light, it is constricted. So kapag ka nasisilaw, lumiliit yan. And then kapag little light or wala masyadong light, it's dilated or naka-open siya or bukang-buka. Okay? So this is actually how it looks like kapag uh, uh, if you check on the eye. So you have your ciliary body or the ciliary muscles around, and then you have the suspensory ligaments that holds your lenses. Okay. So for the iris, so the circular smooth muscles of the iris constrict the pupil. So ganyan kapag constricted, pero kapag nagdadilate, okay, ganyan yung itsura niya. Lumalaki siya. So last, you have the nervous tunic, which is the innermost tunic. It has the retina. It covers posterior 5/6 of your eye, and it contains two layers. So you have the pigmented retina, which is the outer layer. It keeps light from reflecting back in the eye. And then you have your sensory retina, which contains the photoreceptors you call the rods and the cones. So they also contain what you call the interneurons. So for the rods, it is a photoreceptor sensitive to light. There are 20 times more rods than cones, and it can function in your dim light. Okay. And then you also have your cones. These are photoreceptors that provide color vision, and there are three colors like blue, green, and red. Okay, if you check on the picture right here, why is it that they are called rods and cones? Yung shape kasi nila, guys, like this one, the rod, para siyang rod, talagang rod shape siya. Okay, you see here the synaptic endings. It has an axon, a nuclei, and of course the inner and outer segments and the disc. Okay, but for cones, yung dulo niya kasi it's patusok. Okay, it has an axon, a nuclei, and of course the disc. So the pigments and pigment proteins. First, we have rhodopsin. So this is a photosensitive pigment in the rod cells. And you also have the opsin, which is a colorless protein in the rhodopsin. You also have the retinal. It is a yellow pigment in rhodopsin, and it requires vitamin A. So this one is how your retinal cones would actually look like. Okay, or retinal rods and cones to look like kapag they are seen under the microscope. So ganyan siya, mas marami talaga si rods compared to your cones. And then you will see here that on the disc, okay, dun sa outer segment, inner and outer segment, okay, before the disc, you will see here how your opsin and retinal are found. Okay, so in the foldings of the outer membrane of the disc, you will see here your disc membrane. So you will see outside of the disc membrane and then the inside of the disc membrane. Okay, so in the disc membrane, you will see there the opsin and the retinal, which are the composition of your rhodopsin. Okay, so the effects of light on rhodopsin. So first, it is the light strikes the rod cell, and then the retinal changes its shape. So the opsin changes the shape, and then the retinal dissociates from your opsin. The change in rhodopsin shape stimulates response in rod cell, which results in vision, and the retinal detaches from your opsin. So ATP is required to reattach retinal to opsin and return rhodopsin to its original shape. Okay, so this is actually how it looks like. So opsin and retinal are binded together. Okay, so retinal is attached inside opsin to make rhodopsin. So when light activates rhodopsin by causing retinal to change shape, which causes opsin to change its shape as well. Okay, so 3, it activated rhodopsin stimulates cell changes that result into vision. So cell changes stimulated that result in vision. And the following rhodopsin activation, retinal detaches from opsin. So naghihiwalay sila. Okay, umihiwalay yung pinaka-rhodopsin mo. Okay. And then when if energy from ATP is required to bring retinal back to its original form, and then retinal attaches again to opsin to form the rhodopsin. Okay? So the retina, it has rods and cones that synapse with bipolar cells of the sensory retina, and horizontal cells of the retina modified output of rods and cones. Bipolar and horizontal cells synapse with ganglion cells, and ganglion cells axons converge to form the optic nerve. So how does the stimulus travel along your eyes, going to your optic fibers? Okay, so basically in the image that you see here on your screen right now, the direction of the action potential propagation is coming from your pigmented area of your retina, going to the fibers of your optic nerves. Okay, so basically the stimulus would be coming from the source of light. Okay, so from in the eyes, you will see your again your pigmented retina, wherein you will see the pigment cells, okay, of the pigment cell layer. Okay, so once it has already crossed the pigment cell layer, you will see now the photoreceptor layer, okay, already on the part of the sensory retina, wherein you will see the cones and your rod cells. So from your cones and rod cells, it will then eventually go to the interneurons layer. Okay, so the part of the sensory retina wherein you will see there the outer plexiform layer, wherein your cones and your rods meet up with your bipolar cells and your horizontal cells. And then from there, you can also see there the bipolar layers, wherein there may be a few of the amacrine cells and of course your bipolar cells. And then you will also see in in this layer the interplexiform cells. Okay, so those are the connections or the synapses that you call, yung mga pinagdudugtungan nila, mga synapses yan. And then in the inner plexiform layers, you will see now that the opposite ends of your bipolar cells and your interplexiform cells do meet the synapses now of your ganglion cells in the ganglionic layer. So the end of the fibers coming from the ganglion cells, of course, will eventually go with the nerve fibers of going to your fibers of your optic nerve. Okay, so from there, from the optic nerve, it will then be sent to your brain, and of course, the stimulus will then be processed. Okay. Next, you have your retina. So your retina has a macula. This is a small spot near the center of the retina. And then you will also see there a fovea centralis, which is found at the center of your macula. This is where light is focused to when looking directly at an object, and it only have cones on it. So it has the ability to discriminate fine images. And then you will see also an optic disc. This is a white spot medial to your macula. It has blood vessels that enter the eye and they spread over your retina. Axons exit as the optic nerve, and then there are no photoreceptors found, and this is actually commonly called as your blind spot. So for checking or diagnosing or kumbaga, observing how your retina looks like under by using a ophthalmoscope. Okay guys, ito yung ginagamit ng mga doktor kapag chine-check yung dilation or constriction ng pupils natin, and even checking kung may cataract ka ba or kung normal naman yung vision mo. Ito yung ginagamit nila. So as you see there, you have your fovea centralis, okay? And then the macula, that's found on the lateral side. And then on the medial side, you will see there an optic disc and of course the retinal vessels. Okay? So this is actually, or the image B is actually a demonstration of the blind spot. So close your right eye, hold the drawing in front of your left eye, and then stare at the plus sign, and then move the drawing towards your eye. At a certain point, when the image of the spot is over the optic disc, the red spot seems to disappear. So you may actually try to do this one on your on experimenting about your blind spot. Okay? So for the chambers of the eye, you have first your anterior chamber. This is located between the cornea and your lens. It is filled with aqueous humor and it is watery. The aqueous humor helps maintain pressure, refracts light, and provide nutrients to the inner surface of your eye. And then next, you have your posterior chamber. This is located behind the anterior chamber, and it also contains aqueous humor. And then you also have your vitreous chamber. It is located in the retina region. It is filled with vitreous humor, jelly-like substance, and the vitreous humor helps maintain pressure, holds lens and retina in place, and it also refracts light. So the eye functions much like as a camera. The iris allows light into the eye, which is focused by the cornea, lens, and the humors onto the retina. So the light striking the retina produces action potentials that are relayed into the brain, and light refraction and image focusing are the two main important processes in establishing vision. So when you say light refraction, it is the bending of the light in your eyes, and then you should know the concept of focal point. So the focal point is the point where the light rays converge or nagsasama or nagmimit, and it is also where it actually occurs in the anterior of your retina. So usually in focal points, the object is inverted. Okay, so focusing images on the retina associates accommodation. So this is when the lens become less rounded and image can be focused on the retina, and it enables eye to focus on images closer than 20 feet. So this is actually how it looks like to focus by the eye. So usually in a distant vision, the ciliary muscles in in are actually relaxed, okay? And then the tension in the suspensory ligament is high. 'Di ba nga, if you try to or sumisipat kayo ng mga bagay na malayo, 'di ba? Usually nagkakaroon ng tension sa eyes nyo, and then the lens are flattened. And then on your focal point at the back of it, the images are actually inverted. Okay? And then for near vision naman, ciliary muscles in the ciliary body contract and moving ciliary body towards your lens. So the tension in the suspensory ligaments is low, and of course the lens are thickened. But then, kung mapapansin nyo, the focal point is medyo on the middle part of the eye. Okay? Compared to your distant vision na ang focal point eh medyo malayo, medyo nandun siya sa back of the eye. Okay? So again, in accommodation for the near vision, the lens becomes more rounded, allowing the image to be focused on the retina. So again, the focal point is where the light rays cross. So the more or the farther the thing that you are seeing, kung mas malayo yung tinitignan nyo, okay, the focal point is mas malayo. Okay? Pero kapag malapit lang, the focal point is usually malapit, and then more relaxed yung mga suspensory ligaments. So the neuronal pathway for vision, Of course, you have the optic nerve. So this leaves the eye and exits orbit through the optic foramen and to enter the cranial cavity. You also have your optic chiasm where two optic nerves connect. And you have the optic tracts, which are the root of the ganglion axons. So this one represents, or this image represents how your optic nerve and the optic chiasm and optic tract looks like at the base of your brain. Okay? So kapag chinop si brain, yan, si optic nerve ay ito, yung dalawa. So you have the optic chiasm where they converge or nagmi-meet, and then you have the optic tracts below. And then the thalamus, you also have the optic radiations at the side, and of course the visual cortex at the back. So in this one, it actually shows you the different visual fields, okay, of your visual or the neuronal pathway for your vision. So each visual field is divided into temporal and the nasal halves. Okay? When you say nasal parts of the visual field, ito yung mga nasa gitna, this one, itong dalawa. Okay? Pag sinabing temporal, ito yung mga nasa gilid na part. Okay? These are the temporal parts of your eye field. Okay, so after passing through the lens of the eyes, okay, so these are the lens, light from each half of the visual fields project to the opposite side of the retina, stimulating receptors. Okay, di ba nga, kapag nag-pass na yung light, nag-i-invert yung image. Okay, so from then, the temporal retina, axons from the retina pass through the optic nerve to the optic chiasm. So this is the end of the optic nerve, and then they merge into the optic chiasm. So this is the optic nerve, and then this part here is the optic chiasm. So axons from the nasal retina cross, and then those from the temporal retina don't. Okay, so kung mapapansin nyo, okay, let's check. Ulitin natin. Dito sa part na to, yung lighter color, okay, that is the nasal part, right? And then yung darker color, yan yung temporal part. So, kapag ka bumaba na yung optic nerves, ang makikita nyong magkocross dyan, yung mga nasal parts lang. Okay, so yung lighter color nag-cross sa gitna, okay, but yung temporal part, hindi sila nag-cross. They just stayed laterally, or dun lang sila sa side nila. But the nasal parts did cross. Ibig sabihin, nagpalit sila ng position sa gitna in the optic chiasm. Okay? So, optic tracts extend from the optic chiasm with or without crossing to the thalamus, and then collateral branches of the axons in the optic tract synapse into the superior colliculi of the midbrain. So now, after crossing the optic chiasm, you now develop an optic tract, and then these are the superior colliculi of the thalamus or the midbrain. Okay, so ayan. So after it, the optic radiations extend from the thalamus, this part, to the visual cortex of the occipital lobe. Again, sabi nga kanina, the occipital nerve is actually the only cranial nerve in the brain that actually passes or goes out at the back of the head. Okay? So, the right part of each visual field, or the dark green and light blue, okay? Dark green and light blue, projects to the left side of the brain, and the left part of each visual field, the light green and the dark blue, projects to the right side of the brain. Okay? So kumbaga yung mga yung nasal fields, okay, yung nandito sa gitna. Okay? At saka yung nandito sa gitna, sila yung sa right, and then yung sa kabila, yung light, yung dark green, and then yung light blue, sila yung sa left side. Okay? So, usually pag ang vision, okay, since nag-converge na and nagsama na yung different vision neuronal pathways of your vision. So, kapag ka right monolocular vision, ito siya, sa right lang. Kapag ka left monolocular vision, sa left lang. Pero pag binocular vision, nakakakita yung both right and left. Okay? So, so for the visual field defects, so for example, if you have injuries depending on the level ng injury, okay, for your vision, ano bang mangyayari? So for example, you tend to have an injury dito, sa temporal side ng mata mo. Okay, kunyari naputol yan, nagkaroon ng injury dyan, sa letter A. So, what would happen to you is that you would develop central scotoma. So, ibig sabihin, yung right eye mo, kapag tumingin ka sa isang bagay, yung gitna niya wala. Pero yung nasa outer ng picture or sa outer nung nakikita mong view, meron. Yung gitna lang ang wala. Okay? So, it can also happen to the left eye kung sa left eye naman mangyayari yung injury. Okay? So, next, in letter B, you have the mononocular vision. So, if there would be a damage at the level of your optic nerves, you could lose vision. Okay? May it be on the right or the left. Basta complete injury yung mangyayari, magkakaroon ka ng vision loss. And then, kapag ka bitemporal hemianopia naman, it is on the level of your optic chiasm. So, if this one, di ba nga, nag-cross dyan yung nasal sides of your vision. Okay? So, ang mangyayari dyan, kalahati lang of your both eyes ang magkakaroon ng picture or makakakita. So, imagine if you try to view a certain thing, magkaroon ka ng optic chiasm injury, kalahati lang ng right at left mo yung makikita mo. Okay? So, pagka naman sa level of this one, the G, the D, okay, dito sa optic tracts, and then your G, dito sa may bandang optic radiations, and of course your H, okay, or the posterior part of your lower bank of your brain. Okay, what would happen to you? You would have contralateral homonymous hemianopia. Okay, so yan, D, this one, the G, and the H. Okay, this one. Kung dyan ka sa mga parts na yan magkakaroon ng problema, you will develop this homonymous hemianopia. Contralateral, ibig sabihin one side. Okay? So, pag sinabing one side, ayan, dalawa yung mata mo, pero yung both could actually see something, pero parehas silang isang side. Kung yung sa hemianopia, yung outer nila, yung hindi makakita. Pag sa contralateral homonymous naman, parehong side. Okay? Sa left side. And then, kapag ka naman E, sa bandang E, or the lateral geniculate nucleus yung nagkaroon ng problema, or yung J, okay, yung dito sa mismong part na to ng inyong brain, you will have contralateral superior quadrantanopia. Okay, ibig sabihin, parehas ng side, and then, ang makikita mo ay ganyan, parang pizza. May kagat, may kulang na slice sa pizza. So, the rest, the 3/4 of the vision, or the 3/4 of the thing or the view that you're seeing is present, but 1/4 of it is missing. And then, meron din tayong kapag ka sa F, kapag ka sa malapit sa optic radiations, and sa I, yan, dito sa I, yan, pag dyan ka nagkaroon ng problema sa mga calcarine fissures and optic radiations, okay, near the lateral geniculate nucleus, you can also develop a contralateral inferior naman, quadrantanopia. Okay, and then kapag ka sa K, okay, if the posterior cerebral artery is occluded, so nagkaroon ka ng problema with your cerebral artery, what would happen is that you would develop a contralateral homonymous hemianopia with macular sparring. So, ganyan yung makikita nyo. Okay? It's a half siya, half of the view, and then meron pang kalahating circle. So, mukha kang sumisilip sa susian na incomplete. Okay? Those could actually happen depending on the level of your visual pathway, kung saan magkakaroon siya ng defect. Okay? So, please make sure that you are that you know these visual defects. Okay? And which part can be severed or can be occluded kung ano ang mangyayari. Okay? So next, you also have what you call your myopia. This is nearsightedness. So the image is in front of your retina. So for myopia, it looks like this sa baba. Okay, this one, the myopia. So the focal point, it's in front. Okay, medyo malayo. Okay, yan. For nearsightedness. So masyadong malapit yung masyadong malapit yung pagko-converge ng light. Okay? So kung ito ang normal vision, okay, the focal point is actually reaching your retina. Okay, dito naman it's in front of your retina. Kaya siya nearsightedness. And then you also have what you call your hyperopia or farsightedness. The image is behind the retina. So this is the focal point of hyperopia naman. It's behind. Nasa likod siya ng retina. And then for presbyopia, the lens becomes less elastic and you usually need reading glasses for this type of eye defect. Okay, so like hyperopia, your presbyopia is also, or the converging light, okay, or the image is also at the back of your retina. Okay, lumalagpas. So yan. So myopia looks like this. The focal point is found in front of your retina. Yeah. So B is the concave lens that corrects for myopia. Okay, you need a concave lens to correct this myopia. And then this is hyperopia, farsightedness, and then a convex lens corrects your hyperopia. Okay. So other visual defects include also your astigmatism. This is irregular curvature of your lenses. And glasses or contact lenses are required to correct this one. And then here you see that astigmatism, okay, you have multiple focal points instead of having one. So a healthy eye do actually have one focal point only, but with astigmatism, you do have multiple focal points. And then for colored blindedness, this is the absence or deficit of your cones. And they are more likely happening in males. So 8% of the male population and 0.5% of the female population do have the form of color blindness. Okay? So with this one, usually meron nang na-develop, I think, na eyeglasses, colored eyeglasses, wherein people who are born or who had color blindness could actually already see colors by just using that specialized glasses. Okay. And then next you have your glaucoma. It is the decreased pressure in the eyes and it can lead to blindness. So yan, healthy eyes, your eye should be clear. Pagka may glaucoma ka, of course, the central part of your eye could actually or has blurry kumbaga may blurredness, and then ito naman kapag cataract, yung talagang totally lahat buong eye ay nako-coveran. Okay? So this is the chart to determine color blindedness. Okay? If you're familiar, this is what you call your Ishihara color blind test. So this is actually designed for testing the severity of the color blindedness. Okay? There are different designs that are actually kumbaga used in this Ishihara color blind test to determine what type of color blindedness you have. Okay, you can actually search, may mga tawag sa kanila kung ano yung mga differences of the colored blind test. Sometimes kasi the problem is determining like the difference of the primary colors. Some actually naman have the problem of determining anong klase or ano yung tinitignan nilang bagay. Kumbaga more on ano sila, yung mga images ang problema nila. Okay, minsan naman yung iba colors, yung pag-separate nila kung or ano, parang sila nagkakaroon ng confusion of what color it is. Okay? And then So next is the ear. So the organs of hearing and balance are located in the ears, and each ear is divided into three areas. You have the external ear, middle ear, and the inner ear. So for the external ear or the outer ear, it extends from the outside of the head to your eardrum. So the first part would be your auricle, which is the fleshy part on the outside. And then you will see the external auditory meatus, which is the canal that leads to your eardrum. And then you have your tympanic membrane or the eardrum. It is a thin membrane that separates the external and the middle ear. So next, in the middle ear, you will see there the air-filled chambers. So in there, you will see the malleus or the hammer. This is a bony attachment to the tympanic membrane. Next is the incus or the anvil. This is the bone that connects with the malleus to stapes. And you also have the stapes or the stirrup. This is the bone located at the base of the oval windows. So basically, this malleus, incus, stapes are actually one of, or they are actually the most or the smallest bones found in your body. Okay, so they are found in the ear. So next you have your oval window here. It separates the middle ear and the inner ear. You also have what you call your eustachian tube or the auditory tube. This opens into the pharynx and equalizes air pressure between outside air and the middle ear. And then next you have the inner ear. This has a set of fluid-filled chambers. You have there your bony labyrinth. The tunnel is filled with fluid. It has three regions. You have the cochlea, the vestibule, and the semicircular canals. So the membranous labyrinth, it is inside the bony labyrinth and filled with endolymph. Okay, so this is a bigger image of from the external to the inner of your ears. So the inner ear has endolymph. This is a clear fluid in the membranous labyrinth. You can also see a perilymph. It is a fluid between membranous and the bony labyrinth. You will see also the cochlea, which is a snail-shaped structure where hearing takes place. So you will see also the scala vestibuli in the cochlea. This is filled with perilymph. The scala tympani, it is also in the cochlea, filled with perilymph, and the cochlear duct, it is found also in the cochlea and filled with the endolymph. So you also will see there the spinal or spiral organ in the cochlear duct. It contains the hair cells, the tectorial membrane that vibrates against hair cells, and then the hair cells which are attached to the sensory neurons that when bent produce an action potential. So the vestibular membrane, it is the wall of the membranous labyrinth that lines the scala vestibuli. And then the basilar or basilar membrane, this is the wall of the membranous labyrinth that lines the scala tympani. So in this in this image, you will see here that the image A is a bony labyrinth. The outer surface or the gray part is the periosteum, lining the inner surface of the bony labyrinth. Okay, so you will see there the different parts, the semicircular canals, the cochlea, the oval windows, the vestibule, and then the round window, and then the apex. So in its cross section, you will see there the scala vestibuli, which is filled with your perilymph, and then you also have the basilar membranes. So in a bigger picture, the basilar membranes look like this. You have the supporting cells, and then you will see the hair cells. Okay, so the hair cells do have your microvilli, and then the body is of course the hair cell itself, and then it is directly connected to the nerve endings of your cochlear nerve, and then eventually this cochlear nerve um, would exit through your spiral lamina. Okay, and then eventually action potentials would be delivered from your cochlear nerve going to your brain. So how do we hear? Sound travels in waves through air and is funneled into the ear by the auricle. The auricle through external auditory meatus to the tympanic membrane, and then the tympanic membrane vibrates and sound is amplified by the malleus incus stapes, which transmit sound to oval window. So the oval window produces waves in perilymph of the cochlea. And then vibrations of the perilymph cause vestibular membrane and endolymph to vibrate. Endolymph cause displacement of the basilar membrane, and then movement of the basilar membrane is detected by the hair cells in the spiral organ. Hair cells become bent and cause action potential, and it is created. So again, this is actually how the sound waves moves and eventually creates action potentials to be sent to the cochlear nerve. Okay? So this image now shows you the neuronal pathway for hearing. So coming from your from the ear, of course, the cochlear region, and then the vestibulocochlear nerve, it travels now to your cochlear nucleus, found on the base of your brainstem, and then other brainstem of the synapses converge on the other brainstem of the nucleus, and then travels along, okay, to your inferior colliculus, and then the axons from the inferior colliculus project to the thalamus, and then the thalamic neurons project to the auditory cortex. So for balance and equilibrium, first you have static equilibrium. This is associated with the vestibule. It evaluates position of the head relative to your gravity. And then you also have your dynamic equilibrium, which is associated with the semicircular canals, and it evaluates changes in direction and the rate of head movement. So the vestibule in the inner ear, it contains utricle and ursacule. You can also see there the maculae, specialized patches of the epithelium in the utricle and saccule surrounded by the endolymph, and they contain your hair cells. And then you will also see their otoliths. These are gelatinous substances that moves in response to gravity and attached to the hair cell microvilli, which initiate action potentials. So here, these are the locations and structures of the macula. So again, the vestibule includes your utricle and your saccules. So you have here your utricular macula and of course the saccular macula. And then this one, the image in letter B, this is an enlargement of the utricular macula showing hair cells and your otoliths. So the otoliths are the gelatinous, are embedded in the gelatinous matrix. So whenever movement is made, of course, the hair cells would actually respond to it. Okay, and then eventually the hair cells would send the signals going to your nerve fibers of the vestibular nerve. Okay. So the functions of the vestibule is for actually maintaining balance. So in this picture, letter A, in an upright position, the maculae don't move. As you see, it doesn't. The microvilli is not bent at all. So when the position of the head changes, as when the person bends over, the maculae responds by moving in the direction of the gravity. So in this one, in letter B, the picture in letter B, the force of gravity affects the movement of the your hair cells, or of the hair cells. Okay. So semicircular canals are also responsible for the balance and equilibrium. It is for dynamic equilibrium. It sends movement if from any direction. You also have the ampulla. It is the base of the semicircular canal. You will also see the crista ampullaris, which is part of the ampulla, and then the cupula, which is a gelatinous mass. It contains a microvilli and float that is displaced in the endolymph movement. So these are the semicircular canals. So the location of the ampulla of the semicircular circular canals are found here in image A. So as you see, here are the ampullae, and then the semicircular canals. You have here your vestibular nerves, and then the cupula. Okay, so in a bigger picture, enlargement of the crista ampullaris showing the cupula and the hairs. So you have here the cupula. Okay, so in the crista ampullaris part, you will see there your um, hair cells. So it has a microvilli as well. And then the bottom part of the hair cell would actually be the nerve fibers of the vestibular nerves. So the function of the crista ampullaris. So as a person begins to tumble, the semicircular canals move in the same direction as the body. The endolymph in the semicircular canals tend to stay in place as the body and the crista ampullaris begin to move. As the result, the cupula is displaced by moving the endolymph. Okay, they move the endolymph in a direction opposite to the direction of the movement. Okay, so the movement of the semicircular canal with the body movement is contralateral. Kumbaga, magkabaliktad sila. So, kung saan papunta yung position ng tao, ang reaction naman of your semicircular canal would be, or the endolymph of your cupula would be against the direction. So for the clinical applications, so these are the representative diseases and disorders for vision, hearing, and balance. So we will get into some of the examples. So just check on this table from your book or from this lecture that will be provided to you. So first we have trachoma. So trachoma, this is an infection caused by the chlamydia bacterium, and it causes inflammation and thickens the upper eyelid. So scarring of the eyelid pulls the eyelashes into the eyes, and then the lashes scratch the cornea and continue to infect and damage the eye, which can lead to blindness. Okay, so letter A actually shows you the normal conjunctiva showing the area to be examined, but with follicular trachomatous inflammation, that's the example that you see in letter B. And then letter C is the intense trachomatous inflammation. And then letter D is the conjunctival scarring. And then letter E is trichiasis. And F is already your corneal opacity. Okay. Next, you have your chalazion and stye. So chalazion is a meibomium gland obstruction. So meibomium gland is actually found at the upper, or it's near your lacrimal apparatus. Okay, so a chalazion is a specific type of lump in the upper or lower eyelid caused by blockage and inflammation of an oil gland. You call the meibomium gland of the eyelids. Okay, so it is also different with your stye. It is what you call your hordeolum. So usually it infects naman the lower part of your eye. Okay, infection of the eyelash hair follicle. Okay. Next, you have strabismus. So with strabismus, this is actually yung parang nakikita nating naduduling or nababanlag. Okay, so normal eyes usually, syempre direct, and then of course the movement of your eyes can be up, down, right, or left. But with strabismus, tulad nito, we call the esotropia. So the eye turns inward. Ito yung naduduling. Okay, so these strabismus issues with the eye are actually because of the muscles of the eye. Okay. And then, kapag exotropia, the eyes turn outward. So, ito yung babanlag. Kapag hypotropia naman, parang yung kabilang eye ay nagto-turn down or may drooping. Kapag hypertropia naman, the eye turns upward. Okay? So, more likely, these are because of the eye muscles na nagkakaroon ng problema. Okay? So, next is the cataract. Of course, this is, ito yung nagkakaroon ng formation, okay, or nagkakaroon ng block sa mata. Okay? So, ito, cataracts are not only seen in older males or females. Sometimes, babies can also, kumbaga, pwede rin silang magkaroon ng cataract, okay, at their own age. So, maganda ang cataract dapat kumbaga pinapatanggal sila habang maliit pa or kaya pa, kasi kapag ka napabayaan na, it may actually lead to blindness. Okay? So next, you have also the tinnitus. Okay, ito yung pananakit sa tenga. So, this is because of the damaged hairs inside your cochlea. So, kaya ka nagkakaroon ng pananakit. Okay? So, lalo na kapag you're exposed to masyadong malakas na sound, o kaya minsan naman kapag nasampal ka ng malakas, okay? So, tinnitus could actually happen. And then next, we have your Meniere disease. So, it is actually because of a bulge endolymph sac. So, namamaga yung mga endolymphatic sacs mo. So, swelling distorts the sound information. So, that actually ends my lecture for the senses. If you have questions or clarifications, just let me know. Thank you.